So how life works is a fundamental question that all of our scientists here at the Institute are asking on a daily basis. Now, typically, our big ideas series usually brings a scientist who will present a snapshot of their work, and then it's followed by questions. But tonight is different. Tonight, we're really lucky to spend the next hour in an in-depth conversation with just one scientist, which means that we can really dive into how big ideas are born, how they evolve, and how they disperse from the lab into the world. So our hope is that tonight you will leave our institute more curious and with a renewed perspective on both life and nature. Now, what we'll be exploring is really one story with many chapters, and I hope we can cover many of these chapters tonight. But it is really about reading and understanding the vast world of genomes that make each living organism on Earth, that from humans, including us, to the invisible worlds of microbes. And as luck would have it, our guest has been at the center of these worlds for a very, very long time. So I would like to introduce to you our speaker tonight, who many of you already know, from his accomplishments are known to the world. But Dr. J. Craig Venter has led actually one of the first efforts to sequence the human genome, helping deliver a draft map of the human DNA that fundamentally changed biology and medicine. He later founded a multiplicity of enterprises, but he later founded the J. Craig Venter Institute, where there he has teams of scientists investigating human, microbial, and environmental genomics, as well as synthetic biology. So after helping crack the human genome, Craig did something really wonderfully audacious. He turned a sailing vessel into a floating laboratory. And I hope we're going to hear some of that tonight. And then after setting off around the world to study the invisible microbial life in our oceans and what that life means for climate, ecosystem, and our shared future, he embarked on synthesizing genomes from scratch and generating entirely synthetic life forms. So we're very fortunate to have J. Craig Venter joining us tonight. So I'm so excited. It's really difficult for me to say this. I'm so excited. But it's really great to have you here at the Stowers Institute. So please help me to welcome Dr. Venter to the stage. Thanks very much. Thank you. I just get excited. Right, I hope you don't mind that I took the liberty of showing a younger picture of you. I think you were in the Galapagos in that photograph.
I didn't have any more hair, though.
So, as I was sitting thinking about what I was going to start the conversation with, there's a lot of possible beginnings for the stories to start. But what I wonder, which I think many people might not know, is that I really would love to start, with your time as a Navy hospital corpsman, where you were treating infectious diseases both in San Diego and later in Da Nang when you were stationed there during the Vietnam War. And I guess what I want to know from that experience is that in what ways did that early frontline exposure to infection and patient care may have shaped the questions you chose to pursue and the way you think about science?
Well, it was the real beginning. And by the way, it's a real pleasure to be here with you. And I've heard about this wonderful institute. It's nice to see it in person. And it's even more impressive in person.
So, that's wonderful. Thank you.
It's, as I said earlier, you've been able to achieve what we've tried to achieve, but you've been able to go further. So it's nice when that happens.
Thank you very much.
So I was a horrible student. I barely graduated from high school. The reason is I have a trait called aphantasia. It doesn't mean I hate Disney movies. It means when I close my eyes, I don't see any pictures at all. So I only think conceptually. And as I'm sure you know, our education system is based largely on rote memorization. Mm-hmm. So people with photographic memories is the opposite spectrum of my type of thinking. And so learning didn't come naturally to me in a rote memorization environment. And so after graduating barely, I moved to Southern California to take up a surfing career. Uh-huh. But this was 1965, and it turns out if you weren't in college and didn't have a student deferment, you got drafted. So my surfing career went away very quickly, and I ended up in the military. It's a long story, but I ended up in the Navy Medical Corps. And my goal was just to get out of the military with some sort of trade where I could get a job. Because I was working in warehouses and just manual labor. But I found that I had a real aptitude for learning medicine and traits and treatments very quickly. So at age 19, I was teaching interns and residents how to do spinal taps and liver biopsies. In the military, you can do, there's no things based on your degree. It's based on the experience you have and what the people supervising you allow you to do, right? And so at age 19 and 20, I was running the infectious disease ward at Balboa Hospital in San Diego, and was introduced to every major type of infectious disease that people were coming back from Vietnam with: malaria, tuberculosis, cholera, you name it. And that was my first introduction to most of them. It's interesting because it did influence what we did later on. It influenced very much our early choice of genomes after, in 1995, when we sequenced the first genome. We were flooded with money to sequence whatever we wanted. And those diseases played heavily in my mind. But it was much more than that. That whole experience, especially a year in Vietnam, turned my life around totally. And I had to start college from scratch after getting out of the military. And wasn't introduced to high-end science till I transferred to UC San Diego, and just fell in love with being able to ask basic questions. I was given my own lab as an undergraduate. Just found the joy of that you could get paid for asking questions about life and trying to find the answers to them. So that whole time was transformative. Probably if I hadn't gotten drafted, I wouldn't have sequenced the human genome.
So there's a connection.
Yeah.
There's a very strong connection. So, you just mentioned the sequencing of the first genome, and I just wanted to show the audience how far we've come. So this is probably the first microscope ever invented. This was by Antoine van Leeuwenhoek, and he was the first person to actually see germs. Okay, I think he had a rotten tooth. He went in there, grabbed some stuff, and put it on this microscope. Now that's in 1676. So it took about 300 years for us to see a complete genome sequence and assemble. And it's the work that you did. And this is from one of the figures of your paper, the Fleischmann paper in 1995, which was really the first complete genome sequence of a free living organism. And this was a major accomplishment, I think, in the biological sciences. So since this is big ideas, I thought that if you could share with us, and maybe explain also the genesis of whole genome shotgun sequencing, that would be a great thing for us to experience.
And by the way, the Van Leeuwenhoek Award is given out once every 10 years. And they give out as an award an exact replica.
And you have one of them.
And I have one.
That's wonderful.
It actually, you look through it, you can actually see the microbes in it. So I think I'm the only living recipient of that right now. So it's
well, congratulations.
Yeah, so it's nice. But it was a long story. So my work started, most people study something they're interested in. So I was studying adrenaline, because I was an adrenaline junkie. And so I was curious about it. Nobody knew at the time, in the early 70s, how it really worked on cells. So my early claim to fame was proving that it was a cell surface receptor that adrenaline worked on. And then I spent almost the next 10 years of my career trying to isolate that receptor. And realizing that molecular biology was going to be the only route to go to do that. I got invited to move my whole lab in 1984 to NIH to the intramural program. So I took a year off and taught myself molecular biology. And we cloned one of the first neurotransmitter receptors. Bob Lefkowitz was 6 months ahead of me, and he got the Nobel Prize for it. I said, it's his fault I sequenced the human genome because I couldn't compete with him. The early methods, as you know, from sequencing DNA involved a lot of radioactive P32, and it was a very subjective kind of test.
Yes.
You had to sort of guess what the sequence was on the bands. And one of the things I noticed is if somebody published the sequence of a gene, when people resequenced it, they never found anything different than the one that was published. Because it was so subjective, they used, if somebody published something, that must have been right. And so in the late 80s, when Lee Hood and Mike Hunkapiller described the first automated DNA sequencer, I was just fascinated with it. I was just thinking of sequencing more neurotransmitter receptors. And so I used the money that I had at NIH to get the very first machine. Turns out a lot of people had bought them, but I was the only one that get it to work. And published the first paper sequencing 2 receptor genes with it. But in 1985 is when Delbecco and a few other people suggested for the first time sequencing the whole human genome. And my rationale was very simple. I just spent a decade trying to get one protein. So the idea of getting all of them in 15 years seemed like such a fascinating approach that I just immediately became interested in it. And I had the only technology that was working that seemed like it might be useful
Get you
for
there.
that.
Yeah.
And those of you who have done cloning of any genes know you get a radioactive probe and you screen cDNA libraries, which little colonies, about 1 million colonies on a Petri dish, to find the one that has the gene of interest. It was such a slow, painful process to do that and just to sequence the one gene that it's where natural curiosity comes in. I thought, well, what are all those other 1 million spots on the plate? And so I came up with the idea of just randomly picking them and using the automated sequencer to sequence them. And everybody thought it was a stupid idea until it worked.
Right.
Basically every sequence discovered a new gene. And it immediately caused a lot of controversy because I was publishing papers initially with several hundred genes and then with several thousand human genes. Professors from Harvard and MIT and other major institutions would write to call the NIH director and try and get my research stopped because it was unfair that I didn't have to work hard to make these discoveries.
You didn't have to work.
And that every paper I published wiped out a Ph.D. thesis of students that had been working for years to try and get that sequence. Yeah. So my first major idea, instead of being heralded as a breakthrough, was attacked as sort of intellectually cheating by making it too easy to do things. Yeah. But how it led to the shotgun was we had so many sequences and virtually every sequence was new because the databases had nothing in them at the time.
Yeah, that's right.
So we developed an algorithm to assemble the sequences to see what the redundancy was amongst them. And it was actually, we thought, the Secretary of Health asked me at my lab to sequence the smallpox genome as part of an international treaty. So there were 2 stores of smallpox left, one at the CDC and one in Russia. And the idea was once the genome was sequenced, those stores would be destroyed, so smallpox wouldn't exist anymore. And I actually wanted to try the algorithm with the smallpox genome to shotgun sequence it. But the CDC would only give us sections at a time, so we would never have
the full thing,
at one time in the lab, and every clone had to be accounted for, and they would come and take those away before we got the next one. But I encountered at a meeting in Spain Ham Smith, the Nobel laureate who discovered restriction endonucleases. He got the Nobel Prize in 1978, and we just hit it off immediately and spent time that night in a bar and had dinner together and talking about things. And he suggested, he goes, why don't you just sequence a genome? And he said he had the perfect one for us. So Haemophilus influenza is where he discovered the first restriction enzymes. And he said he would make the libraries for us, and we'd use the algorithm and approach that we had, and so that's how it started.
Yeah, no, that's wonderful. Just so you get a sense of what this means, this was the first draft of a genome that's decorated with all the genes that were found in the genome. And so this was part of that first paper. So it gives you a sense of the power of this particular technology had to really advance our understanding of genetic processes. And of course, this led to bigger things, which is, of course, the sequencing of the human genome. I'm just showing chromosome 1, because I think it's the prettiest one in your original publication. But so if you could just take us back, to the 1990s when, there was a public effort to sequence the human genome, which was the Human Genome Project. And then there was your effort, which was a private effort to sequence the genome. And I recall being a graduate student at that time, and I recall, Bill Clinton calling it a tie, which, I'm not so sure it was, but that's what he called it. But once under the lens of history, I'm just going to ask you this question. It's like, what do you think would have happened to the Human Genome Project if Celera had not been in the picture, and vice versa? Yeah. What would have happened if there was no public effort and it was only a private effort? You think we would have what we have today? Would it be different? Would it be the same?
Well, they were both essential. Had I not existed, Celera not existed, it probably would have taken 15 or 20 years to get the first draft of the human genome. Norton Zinder, who was Watson's key colleague and in charge of the budget, said that Congress hadn't even allotted money to do the full genome. And the only way they got money was the threat of me beating them. So Congress gave them $2 billion more. So it's the first time, and I think the last time it's happened. I claim Elon Musk owes me a huge favor, because imagine the US government spent billions of dollars to compete with a startup biotech company. And so they clearly learned from that. So instead of NASA trying to compete with Elon and SpaceX, they funded them and collaborated, and now it's possible NASA may essentially disappear, which would be really a horrible outcome. But the ideas, starting with Dobeko in '85, were just really stimulating. It was a complex project, though, and a lot of people were interested in it for very different reasons. I was coming at it from an evolutionary biochemist point of view. Genetic mappers just wanted to find the disease gene of their choice. So it actually started, it's a much more complex story. We don't have to go into all the details, but when Watson came to NIH, he asked to meet with me because he didn't understand how I was the only one who had the automated DNA sequencer working. And it was the first insult that he gave me when I explained, I said, well, it's very simple. The primer that's used in the sequencing had a secondary binding site. And if you didn't have the stoichiometry, which is the exact ratio of the primer to the DNA exact, you just got gibberish because you got two overlapping signals. It never occurred to me not to do it because I was trained by Nate Kaplan, and you learned how to use a spectrophotometer and you knew what quantitation was. And so I explained this to Watson, and he goes, "That explains it, you're a biochemist." He upped the ante on his insults several orders of magnitude after that, and he had the chance. Tom Caskey and I wrote the first genome proposal to sequence the X chromosome using the automated technology. But the genome mappers actually didn't want sequencing to start, and they were sure the automated sequencer wouldn't work, on scale. It was the silliest notion that the complaint was one machine didn't have enough throughput to do everything. And I said, have you ever seen a sewing machine factory? They don't use one sewing machine.
They use hundreds.
I said, let me show you how to double the throughput. And we bought a second machine. And eventually we had 350 of them.
Wow.
But it just became clear that it wasn't going to get done at NIH. I started my institute in 1992. I was pretty sure I was going to have to not participate in the Human Genome Project. And then in 95, when we sequenced Haemophilus, I was sure that approach would work on a much larger scale. But there was only probably two people in the world that believed it, myself and the mathematician Gene Myers, who was working on ideas mathematically why it should work. But NIH had no interest in funding it. But Applied Biosystems started calling me and offering me $300 million to start a company to sequence the human genome. I thought they were jokes, and I would hang up on the people. I thought they were just great calls. Nobody calls you up and offers you $300 million.
Bucks, yeah.
And, but they were developing a new capillary sequencer, and they invited me out to look at it. It was breadboard devices spread out in several buildings, but it became clear it was going to be revolutionary for sequencing. And we did a calculation that it would take roughly 300 of these machines, and we could do it in less than a year. And so they gave me $300 million to start Celera. And it would have been nicer if it had been cooperative, but there's something to be said for competition. I didn't sort of like the outcome. They used the threat of us doing it to get a couple $1 billion from the government to compete. And that wouldn't have happened in any other country. But we wouldn't have been motivated, Applied Biosystems wouldn't have been motivated without the idea and there actually being the notion of getting the human genome done. So it became an interdependency. I think both helped each other. It was definitely not a tie, though.
If you look at the history, it clearly was not a tie. But I think it was a part of my formative experience to see this grand project being carried out in parallel by both taxpayers' money and by private industry, and now we have a genome, right? And
much sooner
than we would have. So of course, you finished the work with the publication of the human genome, and then you could have stayed focused entirely on human DNA, but so what drew you out onto the ocean to study microbes instead? If you have a chance, go to this website. And this website essentially is an interactive map of the trip that Craig and his colleagues took around the globe to prospect DNA, global DNA, to identify new genes and so forth. But, this is not a logical jump. I mean, you've been fighting, with the NIH, you've been trying to raise money to sequence the human genome. One would think that having all this new information in front of you, one would be attracted to mine it further, but you chose to do this instead.
Well, I got fired. So after sequencing the human genome and raising $1 billion cash for Celera, the parent company decided I was not necessary anymore, and I was kind of a pain in their ass. So I took several months and fulfilled the dream of sailing my boat down to the Caribbean and hanging out for a while I rethought things. And I always had the dream of sailing around the world. It was 2003, a paper was published in PNAS by this very noted group of molecular biologists talking about how little biological diversity there was in the ocean. And that just didn't make any sense to me. It just seemed illogical. And so I thought of an experiment. We were applying shotgun sequencing. We did the first microbiome sequencing from the human microbiome that same year. And I thought, what about just taking a sample of seawater and sequencing the DNA from all the organisms in there at the same time? And if diversity is really low, it'll be very clear cut. So I like sailing to Bermuda, so we went to Bermuda, and we took tanks and had 200 liters of seawater. We filtered them through 0.22, 0.45, and 0.3 micron filters. So we reduced everything in seawater just to these filters that we then froze and took back to the lab and just scraped everything off the filters, isolated the DNA, and sequenced everything at once. And the results blew our minds, and it was probably the paper that changed more thinking than almost any other paper we had. So species by microbial oceanographers were being numbered one at a time as they discovered them. So they're up to SAR 11, which was Sargasso Sea 11. It just didn't make sense to me that all the photosynthesis and half the oxygen we get came from
just
a handful of organisms. So just from this one 200-liter sample of water, we stopped sequencing after discovering around 1,500 new species, and we had 1.5 million new genes that had never been seen before. And we could have just kept sequencing more and more on that, but it totally proved the point that diversity was much greater. But oceanographers usually like sampling the same site over and over again, which makes no sense to me. I swim, I dive, I sail in the ocean, and water's moving
all the time.
Doing a fixed geographic site on land maybe makes sense, but in the ocean, it's like taking a sample out of a river in the same place and thinking you're measuring the same thing. Right? And so I was curious, if we measured something 200 miles away, would it be the same or different? And I reasoned that there would probably be changes in, even if it's the same organisms, there'd be changes in the ratios, where ones that were really rare in Bermuda might be more abundant in different water. Bermuda's right at the edge of the Gulf Stream, and so it's very rich, warm water. And so I came up with the idea of, and it wasn't a novel idea, so the very first scientific ocean expedition was the Challenger expedition
that's right.
in the late 1800s. And at the time, nobody knew what the bottom of the ocean was like. Transatlantic cables were being laid and nobody was sure what was down there. But it was one of the brilliant ideas at the time that there couldn't be any life below a certain depth in the ocean.
Was it 80 feet or 800 feet?
It was
like
amazingly
that, right?
shallow.
Yeah, it's a very shallow number,
yeah.
right?
And so the Challenger expedition took out and stopped every 200 miles and sent a dredge down to the bottom and pulled up things. And every place they did this, including discovering the Mariana Trench, they found life at the bottom and new organisms. But they didn't have a microscope.
That's right.
They could only look at macroorganisms. So my wife hates this term, but you were describing the same thing. Science was limited by visual acuity.
Yep.
And so when we went to the Galapagos, we measured the same sites and everything that Darwin did, but we had tools to see. He was limited to his eyesight. We had the tools of DNA sequencing to look at the same sites again. So we just chose to follow the same route as the Challenger. So you might ask, why 200 miles? The sailors here would know the answer. That's roughly how far a good sailboat can sail in 24 hours. And so we'd stop
every 200 miles.
Every 200 miles and filter 200 to 400 liters of seawater. And we had the only sailboat probably with a minus 80-degree freezer on board. And you can get a lot of filters in a freezer. Whenever we reached a dock, we would FedEx the samples back to the lab where we'd start sequencing them. And my friends in the Department of Energy were very angry with me because I was out sailing around the best sites in the ocean, going scuba diving, surfing, and they were doing the same kinds of experiments in acid mine runoff.
Slightly different environment.
Yeah. So it was, they think my brilliant idea was doing it on my sailboat, not the sampling idea. But we discovered more new organisms than the rest of the history of science put together.
Yeah, I put two of your papers that appear in PLOS Biology reporting some of that trip. I mean, you know, for those of you who have not had a chance to read the book, we have free copies out there. I would encourage you to take a look at it because it really is a fascinating read, especially a journey of discovery. But, you know, since we don't have a lot of time, but if you could take all of us to one specific point that really caused a great impression on you during this trip, where would you take us?
So there were two places that really had a profound impact, but I'd say visiting the Galapagos. I had the rare experience of having my own sailboat there for over 6 weeks, just sailing around there. We had to have a Ecuadorian guard on board and a local naturalist, you know, from the Darwin Society there to make sure we weren't stealing samples or doing it, you know. But it was just fantastic repeating history of visiting all the sites that Darwin visited, basically seeing all the same things he saw because they kept it fairly pristine. It's getting a little overpopulated and ruined a little bit, but with his books and going through the logs and things, and yet doing a totally different sampling and seeing things that he couldn't even imagine were there. But it was all based on the notions and ideas that he created. But it was very adventuresome. One of the sites, I had to take a pump down to the bottom where a hot sulfur gas was bubbling out of the bottom of the ocean. It was by one of the volcanic vents, and sampling right from the middle of one of those. So it was adventuresome as well as just doing science. Yeah. And as you know, we got arrested a couple times, and so there's a lot of adventure other than just taking samples and sequencing them.
Yeah, I mean, you should read the book. It's a great read.
There's audio versions available who like to listen to books and drive on Amazon.
Now you have no excuse, okay? So reading or hearing.
Somebody will read it to you.
3 ways to get it. So, it's I think you also mentioned in the book, so I really want to open the floor for questions. The audience is really chomping at the bit. But I want to ask you one last question, and I'll skip all the other slides I have for you. But it has more to do with, the future of biology. Because as you and I were discussing earlier, I think we're in a really interesting inflection point where for the first time in the history of biology, we have technologies that allow us to see farther and further than any other time in the history of the discipline. And so there's a lot of new biology to discover, a lot of new principles to discover. And in your book, I think you wax a little bit poetic about this transformative experience of being in the Galapagos, seeing the same biology, but now being exponentially complexified much more by the fact that you're going to be sequencing a lot of genomes. And you were, surmising that this complexity is so extraordinary that it might be very, very difficult to really wrap our brains around it. That was several years ago. Being here today, do you still feel that way, or do you think we have a chance to really be able to figure out how life really works?
Well, there's 2 parts to it. We discovered more microbes in the oceans, not to mention probably an equal or greater number that lives in the subsurface of the Earth. More microbes in the ocean than there are stars in the universe. So that's a big number.
It's a big number. It's 30 zeros at least. Yeah.
And then you have to add one more zero because there's 10 times as many viruses as microbes. So there's roughly 10 viruses for every microbe that's out there. And the diversity is infinitely changing. Evolution is constantly happening, which is why instead of all these being nice, simple punctate organisms, I've described them as clouds of organisms with 10,000 related, it's not even clear we can call them species, but variations on a theme, because UV light and oxygen are the main mutagens. And so all the photosynthetic microbes are right there near the surface of the ocean, producing oxygen and living off of UV light. So they're constantly mutating and forming new forms. I don't think we can do it without some of the more advanced computing that's about to be developed. We're talking to OpenAI about doing a major collaboration about just comparing, because we're back to sequencing human genomes, and trying to compare complete phenotype back to the genome. And they said it's going to be 5 years before they'll have computers powerful enough to help me answer the kinds of questions I'm trying to answer. Process all that data, right? So, but it's coming, and it's going to happen. I think the new tools help us reduce complexity by expanding what we can do with our own imaginations. But it's interesting, people overthink what AI can do. I think it's going to be very critical, but for example, with the minimal genome, a third of the genes are of unknown function. We just know if you take them out, you don't have life. AI can't solve that for us. That's right? Because AI can only do things that there's training sets for. So we have to get the answers
to be able to train it.
To be able to train it to get to the next stage. But I think all that's going to happen now 10 times faster than we ever imagined it could.
I'm glad that we share that optimism, because I really think that this is going to be possible sooner than we think.
Well, that's why I'm determined not to retire for at least another 20 years.
All right, well, that's good for you. It gives me hope. All right, we have members of our institute with microphones here, and I would like to open the floor for questions. I'm sure many of you would like to ask Craig a question or two. I see hands already rising, one over there and one over there.
Yes, Dr. Bender, I'm very interested in you're actually seeing face-to-face the complexity of the origin of life, and I wonder how that instructs your inner life.
Well, that's a good complex question. So I'm not sure we've zeroed in at all on the origin of life. I was just talking to the new SIO director, is a planetary scientist, and was responsible for sample return from Mars that we described we could do with our biological teleporter. Earth and Mars exchange about 100 kilograms of material every year. So to me, the biggest surprise would be finding no life on Mars.
On Mars, yeah.
Basically, every time we look at meteors, we find amino acids, including essential amino acids and nucleic acids. Mm-hmm. I think, the Occam's razor approach would be that life started here from panspermia, from some other place. But if I was going to create a bumper sticker, it would just be life happens when you get the right components together. And much in the same way that ideas happen when you have the right stimulants together. Trouble is, some of these distant planets are so far away, it's not going to be easy to answer the questions. Mars will probably be answered first, but if we find, when microbes are found on Mars, they're going to be the same as what we find here, because statistically you can't take a shovel of Earth and not have a Martian microbe in it from all the material that's exchanged back and forth. So I think we need to broaden the scope of our horizons a little bit further. Which means I don't know how to answer your question.
There's another question over here.
Yeah, thank you for a nice talk. I'm a graduate student from KU Medical Center. I like your big ideas and excited to get your book. And I have a question. So unlike math and physics, they have rules and can do some calculation or prediction even. For sequencing for the genome, we need to sequencing species by species, one by one. And currently, with the help of AI, do you think we can train them to make them calculate, summarize rules, and even make some predictions of the species genome during the evolution history, like they used to live on Earth, rather than we need to sequence them one by one?
Yeah. So, mathematical algorithms have been absolutely essential for virtually everything I've done in my career. I did not write a single one of them, and I don't understand, other than a high level, what any of them do. Now, I'm really good at hiring great mathematicians to work with me. As we sequence more and really start to get a really complete look at genomes, the answers are starting to come out very different than they were initially, right? Because comparing humans to great apes, initially we were told we were only a percent or two different at most, but it was by comparing nearly identical regions to each other. Now it's more like 10 to 20% differences. My two parental chromosomes differ by 0.3% from each other. And so we're at the very early stage of still collecting and understanding this information. When asked recently, because it was the 25th anniversary of the announcement at the White House of the first human genome, I said I was extremely disappointed how little has been accomplished in the last 25 years. But if we really are in this new era with new tools, new sequencing tools, new analytical tools, yes, we should absolutely be able to extrapolate backwards at some stage. But as you might know, we made the first minimal genome species, at least the first human-made version. Is probably not the same as an early primitive organism that might have just been based on RNA systems. So I think it's going to be easier to predict the future than to predict the past.
So a question over there. Mm-hmm.
Hi. I've really enjoyed the kind of conversation about the amount of complexity and diversity that you found in your adventures. But I also was wondering if you were amazed by the commonality as well within such diverse and complex systems that you're seeing.
Well, the commonality is we share a code across the billions of species on this planet and elsewhere. And it's remarkable how much that code is shared. And I mean, I sort of find it a combination mildly amusing and annoying when people talk about other mammalian species and dogs, for example, and how they share traits with humans. Like these are major recent discoveries, even though humans have been living with dogs for what, 250,000 years now?
Yeah, at least. Yeah.
And all mammals differ from each other very slightly compared to all the other diversity we see. So I got lost where I was going. So
Differences and commonalities between the genomes. But yeah, I mean, I think it is true that, you know, we all share pretty much the same elements of genetic composition. I mean, I think one of the most profound things that I have gathered from all of these sequence genomes is that it didn't have to work out that way, but it did. That there's a high degree of conservation, but that there's also a high degree of divergence. And especially in areas that supposedly don't code for much of anything, right? But that divergence is absolutely instrumental to the functioning and diversity of species, speciation, and the like. And so I want to take back a little bit the question that the first gentleman asked you, because you also brought the notion of the synthetic life. You know, I cannot imagine what it would have been like when you made the first synthetic chromosome, version 1. Then you decide to boot it up to version 3. I think it was called Syn 3.0 or something like that. Mm-hmm. Then you watch this thing actually divide. So if I were looking at this, I would be asking myself, or trying to expand, what does life really mean, right? Because so if you can actually synthesize this thing, do I expand my definition of life? Do I contract it? What do I do with something like this? So I wonder, you know, when you started playing with this synthetic life, what ran through your head? I mean, is it something that you were really expecting, or is there anything that changed?
We were totally expecting it, and in fact, the biggest surprise was, you know, we assumed, like the rest of the scientific community at the time, that most of science and biology was answered. Mm-hmm. And so when we tried to design the first synthetic chromosomes, each of us, we came up with 4 different designs. I had my own design based on the literature of what components are necessary for life. And we made the chromosomes with the gene content based on those designs and that knowledge. Mm-hmm. Not one of them worked. And so the reason that it took us 10 years to do was because we had to randomly add back unknown genes until we could get a living cell.
Right.
So I'd say the biggest surprise to me was even for the one minimal cell, the third of biology essential for life is completely unknown. And every species, you would get a different minimal answer with a different set of unknowns. So the genes of unknown function outnumber what we know in biology probably 100 to 1 right now.
Yeah. So
we went from thinking we knew virtually everything about biology to it's amazing. In high schools, it's probably my most used quote is that we don't know shit about biology.
And that's scientific terms, okay?
Yes.
Yeah, no, you're absolutely right. I mean, Drosophila, 13,000 genes, maybe 3,000, 4,000 phenotypes. And the remainder, we sort of know, but we don't know what they may be doing.
Look at biology is constantly inundated with extremely naive assumptions. Before we sequenced the first draft of the human genome, the argument was, are there 100 or 300,000 human genes?
That's right.
Because geneticists wanted to have one gene for every human trait.
Right.
And people actually thought it was a fluke when we only found 20,000.
I remember that.
But it gets back to the mathematical question. 20,000 is a huge number when you're dealing with combinatorial genomics and differential expression in each cell with different methylation. I mean, in reality, the possibilities are limitless. And whether we get there by trial and error, which is what's happened with evolution thus far, or we actually take over evolution and start trying to direct it, more likely a combination of the two. The sky is virtually the limit.
Yeah, that's right, and that's our future. That's our future. So I see another question over there, Craig, for you.
Hi, sure. Fascinating, especially to me as an astronomer, getting your perspective on Mars. But there's many outer solar system ocean planets that may be more isolated. So I'm kind of wondering if life may have one basic, if there's a better structure, are the four bases the best, or could life be formed by others, and might we be able to find out by comparing with these other worlds? I don't know if a contaminating life form how that would compete with indigenous life on Earth.
No, as you must know, there's been so many debates, particularly on people looking at extraterrestrial origins of life and stuff. Could it be totally different? Could there be a silicon-based system? I'd say the fact that we're finding the same molecules, the same chemistry, basically everywhere we look, that it's hard for me to imagine ending up with a different system. That my life happens is when you have the right combination of the same chemicals and the right conditions. If the chemical composition was totally unique to Earth, which now we know it's not, trouble is these ocean planets are so far away, there's no way other than through optics to try and test what's there, but it'd be lovely to sample them and sequence what's there. It's really dumb for scientists to rule anything out, because every time somebody does that, they're proven wrong. So, I would bet money on the type of life forms we have here are going to be ubiquitous with probably some exciting variations on the phenotype.
One more question? Yes. There's, well,
So I'm curious to
Yeah.
I'm curious to hear your thoughts on. By the way, I love the insight that you've given already. But I know you plan to not retire for another 20 years. But if you could be really young again in like the
What are you trying to say?
what do you think is the next like big frontier for biological sciences, and where would you want to be? Like at the crux of what would you want to be at right now, working and then striving for the next, 60 years of your career working on?
I've had probably the most unique opportunities because I've been able, all the things that I've done have been questions I wanted to get answers to and things to work on. You can tell I have trouble focusing, because, there's this theme around all these same things of, life being driven by a code and trying to understand that code. So even after 20 years, nobody has repeated what we have done with making the first synthetic cell, because the problem is booting up a chromosome. Mm-hmm. So I set up two teams from the start, one to actually synthesize the DNA and develop the tools for writing the genetic code. But I knew from the beginning the problem was going to be how do you boot up this code? Yeah. And we lucked out with a system that actually a French postdoc brought in of two microbes that the chromosomes could be transferred and the species would transform based on moving the chromosomes. And that affected our choice. But, I think the most fascinating thing that will enable huge amounts of creative change where, the high school science contest will be making a new species, and who makes the most interesting species that has the, or what we've argued is adding things back to our minimal cell, you should be able to completely recapitulate all of evolution. This has happened in baby steps by group added, so the JCVI 3.0 was basically a large blob that pinched off new cells, sort of like soap bubbles did, like Jack Sosak's bubble dividing. And so we worked to add genes back that would restore normal cell division. But adding the known genes that were involved in cell division didn't work. Mm-hmm. We had to add 2 extra unknown genes by trial and error to get there. But so I would probably focus on one of 2 areas or do what I'm doing, focusing on both, trying to create more interesting life from scratch, and better understanding humans so we can make better humans from scratch than we have.
Well, one last question from a new human over here.
Sorry about that. I really loved your lecture.
I don't think it's on.
Yeah, he's.
Much better. Oh.
Yeah, it's working now.
Okay.
I really loved your lecture, and I want to ask you a question. How, when you first began to research, you synthesized so many genomes that people, other scientists, were saying that, oh, he's not working hard enough, he shouldn't be getting all this credit. And I was wondering, how did you respond to these criticisms from other scientists, especially when you were just getting into science?
I didn't understand.
Could you repeat the question again, please?
You're very soft-spoken.
Yeah, speak louder into the microphone.
Sorry about that. I just have a question on, when you were first into science, you were synthesizing so many genomes, and other scientists were just criticizing you for that you weren't getting enough you weren't working hard enough to get these things.
Ah.
And I was wondering, how did you respond to this criticism, especially as a young scientist?
When you believe in yourself and believe in your own work and you understand the importance of it, the criticisms were more like just surprising to me that, you know, of how dumb some people were, right? I mean, I didn't take them personally. You know, it was like, really, a Harvard professor is going to complain because somebody went 10 times faster than they could think of, and it's like, no wonder science is moving so slowly. But the key, you know, doesn't matter whether, you know, you're going to be want to be an Olympic champion or do science. You have to believe in yourself and your ideas. Most of the ideas that I've had for a long time, I was the only one who believed them. When I was offered the $300 million to start Celera to sequence the human genome, I went back to the Institute and told Ham Smith that I had to leave and do it. I was worried about telling this story. Because Ham died a month ago.
Sorry about that. Great.
But his comment was, "I don't think it will work, but I'm going with you." And so you have to believe in your ideas, or other people won't.
Yeah.
It also helps to be right.
Well, Craig, thank you very, very much for your time with us.
you.
Let's thank Craig for his time.
Thank you.
Thank you, Craig.
Enjoyable conversation.
Pleasure.
Yeah.
The conversation will continue. We have a reception in the galley, and please mark your calendars for our next big ideas, which is going to happen in April. And so we look forward to having you here. And Craig, thanks again for a wonderful evening.
Thank you for having
Greatly
me.
appreciate it.
Yeah.
Thank you. Thanks very much.